Okay, good morning, everyone. Again, my name is Fabian Tobar. I'm from Cali, Colombia. And today I will present this project about some initiative in Colombia about precision medicine. In Colombia, now we are working in the applying of some protocols about precision medicine. And this is some of the research project approved the last year with the idea to start in this way in the definition of new protocols about precision medicine in Colombia. And the idea today is to show you the most significant elements about this project and the research idea and what are our preliminary results. Well, first it's important to define what is the main objective in Colombia about precision medicine. The last year was presented this grant in our country to select some research lab in Colombia with the idea to improve the methods of analysis of the information related with the patients, from omics data to clinic data to another kind of information that could be used to the attention of the patients, but with most direct attention or most direct protocols in the attention of the patients. Of course, our project was selected in this grant, and the idea with this project was to analyze specifically the rare diseases, because we have experience in the analysis in my university. We have experience in the analysis of the rare diseases, but specifically in the description of new genes for some diseases or the characterization of the population with some genetic counselor, with the idea to support to the families and to the patients about the treatment of the disease. And the idea with the project was use this kind of information that we have in our university with our experience in the analysis of the human genome composition. Specifically, this is my field. I work in the human genome analysis, specifically in the identification of regularities or properties in the organization of significant elements in the structure of the human genome. And we connect these both fields to integrate a predictive model of association or the genetic association based on structural patterns in the human genome, but apply it to the necessities of several rare diseases in our country. Our project has have 3 phases. We are in the first phase, that is the phase in the development of the machine learning methods for the integration of rare disease information, and of course the analysis of the human genome composition. That is our main objective in these first years. And the idea after that is when we define which are the best parameters for the characterization of the rare diseases by the genomic patterns, the gene compositions, the regulatory mechanisms, the epigenetic mechanisms, we can select some diseases for the experimental validation. In fact, in the next phase of this project, we will analyze or we will study some rare diseases in some population in Colombia. This is a really challenge in Colombia because obviously rare diseases is a disease with a low frequency in the population. Therefore, we need to integrate our public databases with genomic information, and this is a real challenge in Colombia because we don't have these kind of systems, and we are working on that in this moment. And the phase 3 of this project is the validation of the machine learning protocols, and of course, improve the analysis of the diseases, the rare diseases, and the idea is to propose any solution or in the treatment or in the diagnosis or in any field that helps to the patients to solve anything about these kind of pathologies that are so dramatic for the patients and the families. To understand this project with more detail, it's important to present our preliminary studies about the human genome structure and composition. Specifically, I have working in the definition of specific patterns of organization of repeats, DNA repeats in the human genome structure. Repeats, as you remember in the previous conference, are so important for the regulation of the human genome, the regulation of genes, the organization of the chromatin in the nuclei, and other additional functional features. But herein, our main objective was the definition of structural patterns. That means configuration or combination of repeats in specific sites in the human genome. In this study that we published recently, we showed, for example, that the combination or the configuration of specific repeats define functional pathways or functional or significant functional pathways that describe or integrate the genes in a new ways of response to several necessities in the cell. Herein, the most important fact was the identification of combination or repeats in specific sites in the human genome structure and the association with those functional characteristics. And previous to this work, we validate this configuration or these combinations, but applied to some diseases. We analyze, for example, some kind of cancer and neurodegenerative diseases. And we found that there are specific key genes in those diseases with a specific configuration of repeats in the promoter structure. When we evaluate this in a massive analysis with real information from patients, we found, for example, that some of those configurations or some of those combinations are associated to structural variants in the human genome. And that was the first idea, that maybe the structural variants in the human genome could affect the structure not only of the human genome, could affect the configuration or the combination of specific regions in the genome. And that was the support of our main idea for the project. About rare diseases, this is an important, we have experience in the analysis of rare diseases, but it's not at the level of the genomics analysis. In Colombia, we had some ideas about the most significant rare diseases in our country, or we have some experience in the identification of some specific genes related with some specific rare diseases. But one important thing about our experience about the study of rare diseases is that we don't have a clear way to describe what is the most significant genetic factor associated to the manifestation of these chronic or so complicated diseases. Therefore, we work with some genetics in our country to the definition of where are the necessities for the attention of the patient. And our geneticist says that the most significant information that they require is the association of other information not necessarily related with coding genes. They need to know the whole genome composition of those patients to define additional factors, maybe by the epigenetics or something like that. And based on that, we propose that, in fact, with analysis of the configuration of repeats and analysis of the whole genome from some patients, it's possible to identify possible elements in the composition of the genome of those people, with the idea to find if the structural variants in the configuration of those regions could define possible susceptibility regions for those diseases. This is another preliminary study. Here we define, for example, that rare disease-related genes are not rare genes. That means that the genes affected or the genes that are mutated in rare diseases are common genes for other diseases. This is not different. And functionally, there are the same functions for those genes. That means that there is additional factors that describe a rare disease, not only the coding genes, maybe it is related to the epigenetics of or a specific unknown element for the genes, or for the genome of those patients. Here, for example, you can see that when we compare the rare disease related genes with the other genes related with other diseases, functionally in this enrichment, we see the same thing. That means that, as you see in the statement, rare diseases are not related with rare molecular process. Therefore, we need to apply additional methods of analysis, not only related with coding genes, and therefore there is a long way in front of us. About our preliminary findings in our project, now we are working in the definition of where are the best parameters for the classification of rare disease based on structural or configuration of repeats from the genome. Now we analyze, for example, all the rare diseases deposited in databases. There are around 8,000 diseases. And we found, for example, that there is a common association between the functional process related with specific phenotypic traits from some rare diseases with some of our configuration patterns in the human genome. For example, here are just seeing some of those repeats that we have characterized in the last year. And as you can see, there are some cluster of genes related to rare diseases. That means that, in fact, there is a significant role of these repeats in the configuration of patterns or specific patterns for those genes. It allows us to classify or to apply methods of clustering for the discrimination of rare disease-related genes. And in fact, we found that by using this kind of repeats and the configuration of the repeats, that the rare disease-related genes could classify or could define specific clusters. And to each cluster, as you can see here, we can associate functional properties about the cell response to different kind of conditions. Here, we validate the fact that the configuration or the combination of repeats in the promoter structure of the genes is related to the functional characteristics of the cell functionality. But our question today is if these configurations in the promoter region of the genes could be used to describe phenotypic traits for rare diseases and use this information as biomarkers to the a more precise analysis in the genome structure of patients with rare diseases. These are our current questions of research in this moment. We need to define which are the repeat configurations most significant for the definition of sensitive types for a structural variant. Okay, and the other question is, which are the best rare diseases to explore our model of prediction of sensitive types of variation. Well, I'd like to say thanks to my team in Colombia and, of course, all the institutions and centers that support this study in Colombia, and of course, to Rio Bio for support this conference. Thanks.
Thank you, Fabian. Any question to Fabian? Just a quick one. In the rare disease compendium that you have, how many of them are related hereditary disease, and how many are somatic disease, or how do you distinguish that?
You can see here with a genetic association, it's the bigger proportion here. Rare disease has several classifications, but this is not clear information because there are rare diseases and the classification or the description of those diseases in our country or in several countries is not clear. Therefore, there is necessary applied or improve the method of diagnosis to associate any possible molecular or genetic association to each disease. But we are working with the information that we have in our health systems.
Okay, thank you. A quick one.
Hi, I realize you are still at an early stage, but you mentioned a couple times about returning the results to patients. So I was wondering if the process you are aiming to do is like clinically validated to do so, and if you're already working with some genetic counseling team.
Yeah, sure. Yeah, of course, I'm a bioinformatician. I have experience in the attention of patients. We need to work with geneticians in our labs, in our country. And we use the information that they define in the diagnosis or the best treatments to the attention of the patients. And we use all this information to support the idea that what are the specific phenotypic traits that we found in our data that could connect with the information from the geneticists. And we use all this information to define some variables in our method of machine learning to define what are the best candidate disease or phenotypic trait that could be useful for the genetics. Basically, is that.
Okay, thanks.
Thank you.